Hi, hello everyone to the readers of Fertility and Sterility. My name is Marcos Meseguer. I'm a senior embryologist and the scientific supervisor of IBI Valencia, based here in Spain. First of all, I would like to thank Fertility and Sterility to give me the chance to show to this forum and also in your website to do a brief introduction of our paper that has been recently accepted. This paper is the clinical validation of a different way of doing embryo selection and incubation. It has been the clinical validation of a time-lapse monitoring system. It has been performed by a randomized control trial, and for us it has been, let's say, the final work and a summary work of all our last research during the last 6 years here in IBI Valencia and in other IBI clinics, in which we have been doing the introduction of a new technology in our clinic, the IVF clinics. It has been a time-lapse system, the EmbryoScope. We introduced the technology in 2009. During 2010 and 11, we developed a new way of doing embryo selection based on morphokinetics, which basically is the selection of the embryos using the exact timings of embryo cleavages. So once we developed a way to do the embryo selection, what we did was also to take a look to our outcome just by a retrospective study. This study was published in 2012, and we were able to demonstrate an improvement in our clinical outcome of around 10%, 20% relative when we use time-lapse technology in comparison with standard incubation. And once we finished all this research, we finally decided to perform a randomized control trial, which is a lot of work. It was started in 2011-12. We randomized around 850 patients, so it's the first time that any group has performed a randomized control trial comparing a time-lapse monitoring system, which includes a different way of embryo selection, together with a new or better culture conditions, because we don't remove the embryo from the incubator and we keep always in the incubator for doing the culture. So the combination of these two aspects that apparently will improve the outcome were compared with our standard procedure of embryo selection and also our standard procedure of culture in the standard incubations by using also morphology. So more or less, to give you a summary of our results, we take a look into obviously we try to control all the potential bias. These are a controlled randomized trial. The patients were patients of very good prognosis, which basically were not advanced maternal age and also presented a good response with more than 6 metaphase 2 available for ICSI. So they were good patients, and the summary of our results can be simplified in an improvement in our ongoing pregnancy rate close to 10%, also an observation of an improvement in our implantation rate around 8%, and also a clear decrease in our miscarriage rate around also 12-15%. So the introduction of this new strategy has been able to show us an improvement in our outcome, which is a relative improvement of 20%. For us, it has been, I would say, a lot of work, because basically doing a randomization is quite a lot of work. It has been a very large study. It will be difficult to find in the literature a study like this with this amount of patients randomized, and hopefully we will see in the future what happens, but we hope to see more papers and more research in this line to confirm our results, or even to see if there is an option of even improve that increase in the outcome that we have been able to observe by the implementation of time-lapse technology. And that's all. Thank you very much for your attention. I hope you will like the paper, and I hope you will read it. And of course, we are available for any question, and we would like to participate also in the forum of Fertility and Sterility. Thank you very much.